Okay, so today's class is on manipulation of DNA. So the way you manipulate DNA, the various enzymes which are involved, how these are used, etc. So that will be covered in a series of 3 lectures. So first is today, manipulation of DNA. So here we will be knowing a whole battery of enzymes which are used to either cut DNA, join DNA, modify DNA, etc. So before I begin, can you please tell me that if you have to cut DNA, which enzymes are used?
Ma'am, restriction enzymes?
Okay, so in all applications, would you go for restriction enzymes? How about I have to completely degrade the DNA? Then what?
Nucleases. Nucleases, ma'am.
Then what will you use? Are you aware of any enzymes which are used if you have to completely degrade the DNA?
DNAs.
DNAs, exactly. And will all the nucleases, do you think they will work on circular DNA or let's say a linear DNA? Would all nucleases work similarly?
No, ma'am.
I mean, to say that, yes. So today's introduction is to all those different type of nucleases and how do they differ and in which applications you might use them. So before I begin introducing you to various nucleases, so just as I told you that the broad category of enzymes which are used in molecular biology, they are divided into nucleases, ligases which join the nucleic acids, polymerases which make copies of molecules, and again there are so many different types of polymerases. In which application you use which polymerase, we'll come to that, and the various modifying enzymes, etc. So when we talk about nucleases, we talk about those enzymes which have the capability to cut DNA or shorten DNA, and that shortening of DNA, it can be either from the 3' end or from 5' end, or it can be from both ends. And it could be that in if I use certain type of nucleases, it leads to complete degradation of the nucleic acid, and what is the final product is either a single or dinucleotides, etc., or even short oligos, etc. So we'll talk about the various nucleases, and again I want to tell one thing, that when I talk about nucleases, it means the enzymes which have the capability to act on both DNA and RNA. And when I talk about DNases, it means that they are specific only to DNA molecule. And when I talk about RNases, it means that it will only cut the RNA molecules. So again, coming to nucleases, again they have been categorized into 2 broad categories. One is exonucleases and one are endonucleases. So exonucleases are the one which shorten the DNA from the ends. That is, they remove the nucleotides one at a time from the end of a DNA molecule. And again, within this category, exonucleases, we have many other types of exonucleases, and we'll just come to that. But if I just want to means differentiate exonucleases with endonucleases. So endonucleases are the one which break the internal phosphodiester bond, and they do not act from the ends of the DNA molecule. So they act within a DNA molecule. So this difference you should definitely know. So when you talk about restriction enzymes, they are always restriction endonucleases. So in my next class, I'll discuss briefly about the restriction endonucleases, because I think that some of you might have already studied them in your molecular biology course. So coming to this slide, which shows so many different types of exonucleases. Okay, see what has some message on that? Because I can't see. No. So, yeah, coming to, please see this slide very carefully. We have so many different type of exonucleases that have been listed here. Let's see how they differ. First thing that you can see is that the starting material differs between various exonucleases. Some act on single-stranded DNA. Some act on these, they can act on double-stranded DNA, be it the blunt end, be it a staggered end, or be it single-stranded DNA, right? Some require the presence of phosphate group at least on one of the strand at the 5' end. Some do not have that requirement. Okay. Then some can act on double-stranded DNA like blunt or this staggered ends, but some can also act on nicked DNA. Nicked DNA means one strand is intact while one has a nick, or let's say the phosphodiester bond is broken between two nucleotides in one of the strand. So it means that they really differ in the starting material. So as we can go down also, you can see that there are different types of requirements for various exonucleases. So one is the requirement for this starting material. Then is that from where are they cutting? These are exonucleases, but it doesn't mean that they will always cut the DNA from both the ends. That's not the case. So you will see that some have this 3' to 5' exonuclease activity, in which these nucleotides are removed one by one, and some have 5' to 3' exonuclease activity in a double-stranded DNA, or some have this again like 3' to 5' exonuclease activity. So if I, let's say, want to generate a DNA molecule which has 3' overhangs, maybe I will give a brief treatment with lambda exonuclease. So my double-stranded DNA, which let's say initially was a blunt end, so it will finally, after treatment with lambda exonuclease, it will have a 3' extension. Similarly, if I want to have 5' overhangs in my DNA, then I can use exonuclease 3. Obviously, I have to play with the concentration of exonuclease 3 or various exonucleases that I am using, because otherwise it will completely chew up the DNA. So the timing and the concentration of the enzyme that you use will also lead to the final product that is made. Now, what is the final product being made? Like in some cases, it can be certain overhangs, but if you let it work completely, in many of the cases, the final product is either single or double nucleotide, because it breaks after each nucleotide. So you can have, I mean, DNMPs as the final product. But in some cases, the final product that is generally seen is a short oligo. So it does not completely chew it. So all these details are given in a table. So all these figures have been taken from NEB. So one of the major company which is selling these various enzymes, be it restriction endonucleases or be it these nucleases, or be it various molecular biology enzymes that you will come across. So one of the major companies, NEB. I think in majority of the labs in India, you will find NEB products. And unfortunately, we don't have any Indian company which is selling these molecular biology enzymes. And one of you, means any of you who are interested, you can actually open this company, and it's a huge market, because you can understand that in every molecular biology lab, you will have many of these enzymes, and the requirement is very regular for these enzymes. So coming to exonuclease 7, it cuts from both the sides. So it shortens the DNA from both the sides. Then we have nuclease BBL733, which acts on double-stranded DNA or single-stranded DNA, and again acts from both the sides and on both the these strands. Then coming to exonuclease 5, Again, it is cutting the DNA from both the ends, and the final product is a very short oligo that is left in the solution once you treat your DNA with this particular enzyme. So, in my subsequent slide, I've given you two links for the NEB brochure for these enzymes, in which you can see the specific requirement of each of these enzymes. And you will see that some of these they act on linear DNA, some of these they act on nick DNA, some of these they act on double-stranded DNA. You can also see that some of these they have been developed as thermotolerant. So I have told you, and I'll tell you multiple times, that this is a very, very dynamic field. The enzymes that have already been isolated from various sources, even they are being made better by various techniques, by site-directed mutagenesis or random mutagenesis, etc., so that they work better. And the unwanted properties that these enzymes have, if they can be removed by your interfering with these, or let's say by manipulating these enzymes. So all these details are given in that brochure, and also that in which particular applications in molecular biology, which of these enzymes are used, even that is given. So for example, exonuclease 5, what is its speciality? Is the fact that it degrades the contaminating linear DNA. So it acts on linear DNA, but it does not act on circular DNA. So if your plasmid preparation has contamination from linear DNA, that is chromosomal DNA, which obviously will be broken into small pieces, it will not remain, let's say, circular, even if it's a bacterial chromosome. So that can be removed with the help of exonuclease 5, and it will not act on it. There were, can I use for example, exonuclease 3 for removal of linear DNA in my plasmid preparation? No, I cannot use, because it is acting on nick DNA as well. And you can, I hope you know, and I had told you in my previous class also, that when you isolate plasmid, there is a chance that apart from supercoiled plasmid, you can also isolate nick as well as linear plasmid. So if you want your plasmid preparation to be completely circular in nature, etc., then it's better to use exonuclease 5. Okay, so this is just to sensitize you that we keep telling, okay, exonucleases, but how much variety in exonucleases we have. And if you become a molecular biologist and means working in a molecular biology lab, then depending on your application, how do you decide which exonuclease to go for? Then these three are also nucleases, but they are endonucleases. And how do they differ from restriction endonucleases? Is the fact that they do not have much, means they do not recognize a specific DNA sequence, like you know that restriction enzymes, they recognize a specific sequence and then only cut. But these nucleases that I'm talking about, DNAs or MNAs, etc., they are non-specific in nature. But you must have read that they are generally non-specific in nature. But in some studies, you might see that DNase 1 has, certain this certain affiliation to certain bases. So sometimes you'll see that some of these enzymes they cut after, let's say, A, U, etc., or AT, etc. So many studies have been done that are they completely non-specific in nature, or do they have some specificity? So one point is this. Second point is this, that some of these enzymes they also have a certain means affiliation towards, for example, they'll cut more in the minor groove, etc. So a lot of studies, whenever a company develops a particular enzyme or studies a particular enzyme, so before bringing it in market, it goes through multiple checks. They check, okay, what are its various properties, and then they report in the whatever brochure they are means putting on the web. So you can go through the details of those if you are interested. But in general, they, if I go in more layman terms, in general, they are non-specific endonucleases. and that's how they differ from restriction endonuclease. So DNase 1, it makes a cut in a double strand DNA, or means any of these starting materials can DNase 1 can act on those and make a cut in those. Similarly, micrococcal nuclease, it will make a cut in this many molecules. Only difference between this and DNase 1 is the fact that in MNAs, the DNA products, the DNA fragments that are finally made, they have a 5' phosphate as well as, sorry, a 3' phosphate and a 5' OH group. So for further ligation reactions, if somebody wants to use it, those can be used. But that's the only difference between these two, that this generates a 3' phosphate and a 5' OH group. Then coming to S1 or a P1 nuclease, it only acts on linear DNA, and it is very, very useful in certain experiments or certain applications. When I'll be teaching you about the transcript determination, etc., if can you determine the intron exon boundary of a transcript, and how do you determine the ends of a transcript? So in all those cases, S1 nuclease becomes very important. It means that if there is a preparation of a double stranded DNA or a single stranded DNA, it will specifically only cut single stranded DNA. So you can understand that if I'm talking about determining the intron exon boundary, so if I hybridize my genomic DNA or primary transcript with the mature mRNA, so what will happen, that the intron, it will remain single stranded because it will not hybridize, it won't have any hybridized template to be hybridized in case of mature mRNA. So that single stranded DNA or RNA, it can be removed with the help of S1 nuclease. So it is used generally in those scenarios. DNase 1 you will keep hearing when you are isolating total RNA. So in if you are isolating total RNA and you want to get rid of the DNA or contaminating DNA in your RNA preparation, then you generally treat it with DNase 1. MNase 1 is generally used when you are doing some chromatin immunoprecipitation experiments. So in those, generally when you see the various protocols, they have generally used MNase 1. And it was seen that if the DNA is protected by the protein, or let's say by binding of a nucleosome or a protein transcription factor, etc., then MNase 1 cannot act much on it, but the naked DNA is acted very well by it. So how these see DNAs, instead of DNase 1, you can also use MNase 1 in your in removing DNA from your RNA preparation. But various standardizations have been done, and it was found that okay, generally DNase 1 is used for removing DNA. So again, a recap and some examples of the various nucleases that we have just talked about. For example, BAL31, it cuts from the ends. It's an exonuclease and it acts from both the sides. Then exonuclease 3, it acts from 3' to 5' on one of the strand. So it generates 5' overhangs. Then DNase 1, it's an endonuclease. So any double-stranded DNA or even single-stranded DNA, sorry, I think it works only, no, it works on both single and double-stranded DNA. And what it does, it makes a breaks in between. And S1 nuclease is the one which acts only on the linear DNA. So all these points you must remember when you are doing some spatial applications. And these are the 2 links I was talking about. Please go through it just to broaden your understanding of the various types of nucleases and in what different scenario they differ from each other, etc. And what is the spatial application for each of these enzymes. Now coming to RNases. So as the name suggests, yes, these are the nucleases which are specific for RNA, and they make a cut in RNA. So do they have some preference for a particular base? Yes, in some cases. For example, in case of RNase A, uh, I also want to just tell you, which I forgot with respect to the nucleases I just talked about, that the source of these nucleases, they differ. Some are from bacteria, some are from micrococcus, like micrococcal, some are from mung bean. So different, some are from phages, etc. So from various sources, these nucleases have been isolated and further characterized. But does it means will no more nucleases be will be identified in the future? No, as more and new organisms are being identified and characterized further, maybe in future we'll have other nucleases that we will be studying. So with respect to RNases, I told you that yeah, they cut on RNA, and RNase A, it is obtained from this particular source written here, and also it is generally seen that it cuts after C or U. Okay. Similarly, you can see RNase T1, it is derived from Aspergillus oryzae, and it cleaves next to guanosine. So now, generally, RNase A, you will see that in your plasmid isolation kit, or whenever you are isolating genomic DNA also. So to get rid of any contaminating RNA in your DNA preparation, you generally treat your sample with RNase A. This is when you will come across RNase A in various kit protocols, etc. But now I had seen, I was going through the catalog, etc., and I found that now new preparations have been made in which RNase A has been combined with RNase T1, because see, it was cutting after Cu and T1 was cutting after guanosine. So they are saying that RNase A plus T1 preparation, which they are selling, is better than RNase A alone. So, but still in general, I would say that in most of the molecular biology labs, you will find RNase A being bought as compared to T1. Then you again see RNase H. See, so how does it cut? It cleaves the RNA when present in the form of a hybrid, in DNA RNA hybrid. So, you know, you must be knowing what is complementary DNA. So, if you take RNA as the template and you use reverse transcriptase, so what you get is a complementary DNA. So that is called as RNA dependent DNA polymerase, or also called as reverse transcriptase. So once it has made the complementary DNA, what we are left with is a complementary DNA and a RNA hybrid. So if I have to get rid of the RNA in the RNA DNA hybrid, what I go for is RNase H. So now in many labs, they are buying the reverse transcriptases which have inherent RNase H property. So it cleaves the RNA once the hybrid is made. And in case of do we always go for a reverse transcriptase which has the RNase H activity? No. In many cases, you may prefer that reverse transcriptases are required which lack the RNase H activity. So in those cases where you want to prepare and go for longer cDNA, so it is preferred that you use a reverse transcriptase which does not have RNase H activity. So yeah, so A, H and T1. Then RNase 3, you will keep hearing whenever you are talking about the, if you want to process your primary transcripts into smaller transcripts, like in case of microRNA generation, if you want your primary microRNA to be processed into mature microRNA, you treat it with dicer or drosha. Okay, so all these have RNase 3 properties, and also it cleaves precursors to make mature rRNA, snRNA, and snORNA. Then RNase R, very important enzyme for those labs which are working on circular RNAs. So I don't know whether you know or not, but there is a new class of RNAs that have been discovered, which are circular in nature. So they are called circular RNA, and because they don't have ends, so they are considered much more stable than, let's say, other forms of RNA. So they are also known to have a lot of regulatory roles in mammalian cells, and studied extensively in certain labs. So in those labs where you want to specifically isolate circular RNA and you want to get rid of any linear RNA which is present in your preparation, then what you do is you treat it with RNase R. Okay, so RNase R can degrade all linear RNA. Then coming to RNase P, it's a ribozyme RNA that acts as a catalyst and functions to cleave off the ends, or let's say extra bases in the tRNA molecules. So believe me, this is not a comprehensive list of RNases that I've shown here, but yes, some of the important RNAs, especially RNase A and RNase H, you will keep hearing about that in your molecular biology practicals, etc. In if you are making cDNA, or if you are isolating DNA and want to get rid of RNA. So similarly, I would also say that even this is not a comprehensive list. Those who are interested can go, can as a hobby read NEB brochure and keep themselves updated with the various enzymes. So this was all about the various nucleases, and of course what we are missing here today is the restriction endonuclease, which will be which you must know obviously for any cloning purpose, etc. So that will be covered in the next class. Coming to the next category of enzymes, that is ligases. So if you cut DNA, you also want certain enzymes which can join DNA. So that's how you make recombinant DNA, by cutting random fragments of DNA and joining them with your vector of interest. So in those cases, you come across these enzymes, ligases. The two most common these origin of these ligases is from T4 phage, bacteriophage, or from E. coli. The two ligases which are in the market are E. coli DNA ligase or T4 DNA ligase. And in majority of the labs, you will only find T4 DNA ligase. Why you only find T4 DNA ligase? I just come to that. So what ligases do, that they repair the broken phosphodiester bond, that is they make a fresh phosphodiester bond between two nucleotides. So how do they do it? These enzymes, they form an enzyme AMP complex. How do they make this enzyme AMP complex? By so T4 DNA ligase, it is ATP dependent, and E. coli DNA ligase, it requires NAD. So in these reactions, what finally we get is a enzyme AMP, or let's say ligase AMP complex. This enzyme AMP complex, the AMP, it reacts with the broken this 5' phosphate group, and once this it makes a complex with that, then it is attacked by the 3' OH group, and finally we have the phosphodiester bond being made. So it's written here, you can just go through it. So this is how the T4 DNA or the E. coli DNA ligases, they work and make a phosphodiester bond. Now coming to that, what is the difference between T4 DNA ligase and E. coli DNA ligase? So it is found that while both of these DNA ligases, they work very well for joining the DNA molecules which have staggered ends, right? Uh, where do I show? Yeah. So it means, see, this is a blunt end DNA molecule. These are the staggered ends, having either a 3' this extension or a 5' extension. So probably I'll make this more clear in my next class when I'll tell you how restriction endonucleases work. So again, I want to tell you that when you have to join DNA molecules with staggered ends, then you can use any of these enzymes and they work equally well. But if you have to join molecules which both of which are blunt ended in nature, then E. coli DNA ligase is not a good choice at all. T4 DNA ligase is very, very efficient in the same. So if you have to join blunt ended molecules, you go for T4 DNA ligase. And it is only for this purpose that most of the molecular biology lab people, they go for T4 DNA ligase in their lab, because there is no point in buying E. coli DNA ligase when it will be used only for some of the ligation reactions. T4 DNA ligase can be used for both of them, both for joining staggered as well as the blunt end. Now, so this is again a chart from NEB that I have put across, just again to sensitize you that there are so many different ligases which are there in the market. You might have just probably till now you must have been only heard about T4 DNA or E. coli DNA ligase, but there are so many different ones which are in the market, and I can assure you that many more will come in future. And based on the number of stars they have, it is that they are preferred in a particular condition. So for example, T4 DNA ligase, I told you it's good for sticky and blunt ends, right? But E. coli DNA ligase is only good for sticky end. Now, a variation in T4 DNA ligase was made, and a salt resistant T4 DNA or salt tolerant T4 DNA ligase was used, because many times it was seen that when you are putting up a ligation reaction, if the salt concentration is little high, then T4 DNA ligase did not use to work that well. So now they have made a variation of it, which is salt T4 DNA ligase, in which it works very well even in the presence of salt. So some labs actually have started to buy this particular molecule. Then yeah, I think mainly I wanted to tell this, and then you can see there are other T3 DNA ligase, T7 DNA ligase, etc. And they are what use they are put into is written here. You can just go through it. Okay. Then comes that when you are setting up a ligation reaction, what temperature do you use? Okay. So if you have to ligate two molecules, your major job is to bring those two molecules together, right? The more close those two molecules are, better are the chances for them to ligate. If the molecules that you want to ligate, they have recessed or let's say staggered ends, then and let's say compatible ends, okay, like for the fragments which are generated by restriction enzyme digestion, in which you have complementary bases at the ends of the DNA molecule. So in those cases, still the two DNA molecules, they can come together because of complementarity, and the ligation is comparatively easier. But imagine that if it's a blunt ended ligation, then for those molecules to be bound together, there is no force, there is no complementarity in the ends. So in those cases, it's best that you bring the two molecules as close as possible. So that's why you have to play with the temperature that you want to use. So if I talk about that, at what temperature this DNA ligase has the best activity, it is 25 degree. So if I set up a ligation reaction at 25 degree, let's say for 1 hour, maybe in case of blunt end ligation, I will get the ligated products. It could be that depending, maybe if I put this ligation reaction at a lower temperature for longer time, maybe the efficiency was better. But that all you have to play about. And I want to tell you that optimal activity at 25 degree. But if our idea is just to get the two molecules together, then you don't want, you know that in cold temperature there will be not much movement of these DNA molecules, and they'll be more comfortably close to each other as compared to a higher temperature. So should I put my ligation reaction at very low temperature? So yes, I can put like 4 degree or 8 degree have been standardized. So there are various ligation reactions for each ligase that have been prescribed by the company which is selling it. In many cases, you will see that it's a quick ligation, 25 degree for 1 hour is good enough. So yes, it may be good enough for those cases in which you have to ligate two molecules together. So let's say in your tube you have 5,000 vector molecules and let's say 2,000 insert molecules, and you want to ligate them and then transform them, and you want to get your ligated product. So even if 10 molecules have bound to each other or ligated to each other and then you transform, maybe it's good enough in that particular purpose. But if you are making a genomic library or let's say making a cDNA library, then it's extremely important that the ligation reaction that you are setting up is extremely efficient in the scenario that probably all the insert molecules get ligated to the vector molecules. So in those cases, you do not want to take chances and you want maximum efficiency. So in those cases, you will see that all these kits or the protocols that are developed, they generally recommend that you set up a ligation reaction in a very, very small volume. Why do you want a small volume when you are setting up a ligation reaction? Because you want all the DNA molecules to be as close as possible. So even in 5 microliter or 10 microliter reaction, means it is prescribed that you set up in either 5 or 10 microliter reaction. And you set it up at 4 degree for 2 days or at 8 degree Celsius for one whole day. And that is how for library preparation, your ligation efficiency is very, very important. But for your general, let's say one vector, one insert ligations, even 16 degree for few hours or 25 degree for 1 hour, it is good enough in most of the cases. Okay. So this is depending on what is your final application, you can decide what ligation temperature you use and for how long. This slide is just to tell you that believe me, there are some RNA ligases also. You might not have heard till now about those, but yes, there are certain RNA ligases which are there in the market, and they bring together the various RNA molecules and make a phosphodiester bond between two different RNA molecules. So in various RNA-based applications, it might be involved where you want to join two RNA molecules and maybe study its functioning or whatever, some reaction between the two that you want to study, you can do using RNA ligase. Okay, so now coming to polymerases. So as the name suggests, these are the enzymes which make polymers of nucleotides. So again, depending on what template is used, there is a categorization of these polymerases. So there is DNA dependent DNA polymerase. It means it uses DNA as the template and puts the corresponding nucleotide. So it copies DNA into DNA. Then there is RNA dependent DNA polymerase. Again, it's a reverse transcriptase and it copies RNA into DNA. So it uses RNA as the template. And then there is a DNA dependent RNA polymerase which transcribes DNA into RNA. So this is a RNA polymerase. So depending on what it is polymerizing and what it is using as a template, there is various categorization of the same. And there are certain enzymes which don't even need a template. For example, terminal transferases. I'll just come to those enzymes, but they do not need a template for polymerization of the nucleotides. So they just need a 3' OH end and then it will polymerize. Okay, so in case of DNA dependent DNA polymerase, what it requires is the DNA as the template. Because DNA polymerases cannot polymerize de novo, so they always require a 3' OH group to polymerize the various nucleotides which are corresponding to these template nucleotides. So in any DNA polymerization reaction, you will always require a primer, which is unlike the case of RNA polymerases. In case of RNA polymerases, it requires DNA as the template and it does not require a primer. It can just start de novo from here. Okay, so that's the one major difference. So DNA polymerase always requires a 3' OH group, and once it requires, so let's say a DNA polymerase will come and keep adding the fresh nucleotides which are corresponding to these nucleotides. So by which activity does it add these nucleotides? By 5' to 3' polymerase activity. So all the DNA polymerase have a 5' to 3' polymerase activity. Then what other activities they have? If they have put some wrong base, let's say here instead of T, it by mistake, because every DNA polymerase or RNA polymerase has some error rate, and that is documented in at many places and even in the brochure shared by the company. So every polymerase has a some error rate. So let's say it puts a G here. So how it removes it? By its 3' to 5' exonuclease activity. Okay, polymerization is from 5' to 3'. It requires a free 3' OH group, and proofreading activity is the 3' to 5' exonuclease activity. Okay, so this is one important point. Second point is, for example, polymerase called DNA polymerase 1. So this DNA polymerase 1, I think you might have heard about it when you must have been taught about Okazaki fragments. So how do you join the Okazaki fragments and how do you remove the RNA primer in between, etc. So in those cases, DNA polymerase 1 was important. So anyway, so let's say there is a nick, or let's say absence of certain nucleotides in this DNA molecule, and I use a DNA polymerase 1 in the tube in which I want to polymerize and fill in the gap. So what will happen? DNA polymerase 1 will come and add the corresponding nucleotide. But will it end at T here, which is corresponding to A? No, because this enzyme also has 5' to 3' exonuclease activity. Okay, so what will happen? It will fill in the gaps by, let's say, adding C, A, T, T corresponding to these. And here, because it has 5' to 3' exonuclease activity, it will add G, C, A, T, means it will remove the initial nucleotides and keep adding the new nucleotides against it. Okay, so this is how DNA polymerase 1 works. This is how DNA polymerase 3 works. So if I talk about DNA polymerase 1, it has three different important activities. One is 5' to 3' polymerase activity. It has 3' to 5' proofreading or exonuclease activity, as well as it has 5' to 3' exonuclease activity. These you should remember. Now, because it's 5' to 3' exonuclease activity is not required. Here I just wanted to fill in the gap. So now in the market we get clinofragment, means a DNA polymerase in which certain which has been acted upon by some protease, a fragment of it has been removed, and what we are left with is a clinofragment in which the 5' to 3' exonuclease activity is gone. So what will this enzyme do? It will just fill in the gaps. So it will not then start removing these, etc. These existing nucleotides, they are not replaced. So in many scenarios, clinofragment is preferred over DNA polymerase to fill in the gaps or the nicks. Okay. So the another enzyme that is reverse transcriptase, and in this case it is using RNA as a template, and against each RNA nucleotide it is adding this means corresponding DNA nucleotides. So I have already told you about clinofragment, and you can just go through these notes which are written here. But the fact is that it does not have 5' to 3' exonuclease activity, and it is used in many different scenarios in the molecular biology lab. So because it has 3' to 5' exonuclease activity, it can remove the 3' overhangs if they are present in a DNA molecule. Because it has a 5' overhang, it means, see, this is a 5' overhang and recessed 3' ends. So for these staggered DNA, it can make it blunt by filling in. Okay, so it is used for that purpose also, and in many means, as you will learn various different techniques, how do you make probes, etc., you will also understand that the same can be used in generating probes using random primers, or for secondary second strand cDNA synthesis, etc. Now, another variation of clinofragment was made. So, clinofragment, which all properties it has? It has 5' to 3' polymerase, and it has a 3' to 5' exonuclease activity. But let's say in many scenario you don't want any 3' to 5' exonuclease activity. So they have developed another enzyme called exoclinofragment, which lacks any exonuclease activity. Any exonuclease activity. So it only has 5' to 3' polymerase activity. So why is it desired that you don't have the 3' to 5' exonuclease activity? So it is required in certain condition where you do not want to take any chance, and you do not want the last base to be removed in any case. So it is in sequencing reaction that generally this enzyme or this property is desired. And when we go to how sequencing is done of any DNA fragment, I'll come back to this particular enzyme. Okay. So, but again, like for your information, exoclino means it does not have any exonuclease activity. It only has 5' to 3' polymerase activity. So, I think I'll end here, and in my next class, probably I will. Can I get 2-3 minutes more? Yeah, I can. I have. It's 9:48. Okay, so now coming to T4 DNA polymerase. So it is again a DNA polymerase. The only thing is it is from T4 bacteriophage. Okay, it is not the E. coli DNA polymerase that I just discussed in my previous slides. So this T4 DNA polymerase, it has a 5' to 3' polymerase activity, and the difference between this and the DNA polymerase is that its 3' to 5' exonuclease activity is many-fold higher as compared to the this DNA polymerase one. So generally, to remove the 3' overhang, T4 DNA polymerase will be preferred as compared to E. coli DNA polymerase. So if you have to fill in, uh, I mean these two properties, if you want to fill in the this gap, then T4 DNA polymerase works very well. And if you have a 3' overhang, no photo is given here, but let's say you had a 3' overhang, then that can be removed by 3' to 5' exonuclease activity. And I already told you that DNA polymerase, this activity is not that potent as compared to T4 DNA polymerase. So in those cases, you go for T4 DNA polymerase. So if I ask you a question that which enzyme will be preferred for both fill-in as well as remover of the overhangs, then the answer should be that it is T4 DNA polymerase. Okay. And again, from NEP, those who are interested, they can go through this chart. It tells the individual error rate of the various polymerases which are listed here. And as you can see, the exoclino, because it does not have any exonuclease activity, especially the 3' to 5' exonuclease activity, its error rate is very, very high. Okay, but yes, in certain situation, it may be required for having this exoclinofragment, like in sequencing reactions. And then you can also go through that what are the resulting ends of these polymerases. So you will see that some polymerases, they leave a blunt end, some leave a 3' A overhang, etc. Okay. I know it's a bombardment of so many enzymes, but as a molecular biologist, you have to know all these various enzymes, and because then only you will be able to plan your experiments well. Because in your particular application, what combination of enzymes you require is very, very important. Coming to tag DNA polymerase, we will revisit this when I'm telling you about PCR, but I'm probably 100% sure that you know about this tag DNA polymerase. It is a polymerase which works very well at very, very high temperature also, and which is required in any PCR reaction, and it's thermostable. And only difference is that when you use a tag DNA polymerase, it leaves a 3' A overhang. Okay, and I think I'll revisit this when I'm talking about PCR. But one point is that in general, it's seen that it leaves a 3' overhang. And whether all DNA polymerases leave a 3' overhang, no. Some polymerases, as I've told you, they don't have any overhang at the end. Will all polymerases tend to have adenine at the end? No. There are some polymerases which may give a G or a C as the overhang. So it all depends on the properties of the polymerase from where it is, means from various sources that have been derived, and that which sort of overhangs they are leaving. But this property, I will revisit it, that a tag monomerase does leave a overhang when I'll be telling you about the various ligation strategies, and we will revisit this particular uh, fact that I'm showing you here. Okay, so this is the last slide, and I want to tell you that there are various DNA modifying enzymes. There are enzymes which can remove the phosphate group. That's why their name is given as phosphatase. Okay, so like alkaline phosphatase. There are enzymes which are called which add a phosphate group. So they are called as kinases. So polynucleotide kinase. Then there are certain enzymes which add the nucleotides at the end without having any template against which the nucleotides are to be added, and these are called as terminal transferase. So I will end it here, and I will revisit this slide before I start my next lecture on Friday. But I'm open for questions.
Excuse me. So my question is not related to the lecture today, but to the assignments lecture that we saw yesterday. So are we
Harsh, you're
supposed?
not audible to me, it seems.
Now, am I audible now?
Comparatively better, yeah.
So my question is related to the assignment lecture that we had yesterday. Uh, are we supposed to submit anything related to that primer design and real search assignment?
For the primer design, no. So I've not forwarded you the assignment that you are supposed to do. I'll forward that.
Okay, ma'am, sure. And are there any?
Actually, each of you, Harsh, will be given one gene to work upon. For that particular gene, you have to design primers for cDNA. You have to design primer for cloning its promoter. You have to design primers if you were to clone the cDNA with the GFP fusion, etc. So all those means lectures are also there, which you can go through and which will help you in doing a particular assignment that is being given to you. So I'll forward that particular this assignment work to all of you today.
Sure, ma'am.
So please go through the slides. Acha, I have not forwarded you the slides. I will forward all the slides to you today also for both the previous lecture as well as today's lecture, because there are so many enzymes, but they are very, very important. So please go through these slides before you come for the next lecture. And
Ma'am,
tomorrow we are not having any class. We'll have class on Friday.
ma'am,
Huh?
for the ligation temperature, you talk about two things. First thing is that do the ligation at 25 degrees Celsius, where enzyme activity is much more. And
Hmm.
another thing is that use it come back to 4 degree or 8 degrees Celsius, so that
Right.
the DNA molecules come together as
Right. Means
much as.
somewhere a compromise. It might be a compromise in your DNA ligase activity, but still you are giving enough time to it. You are giving like 2 days. So DNA molecules are in very much proximity to each other because you always have to set up ligation reaction in minimal volume. And this has been standardized by various companies and molecular biology labs, and then these protocols have been developed.
So like we will be going with 25 degrees Celsius.
See, in your regular thing, if you're not making a genomic or a cDNA library, and in which you have to have excellent efficiency, you don't want to miss on any DNA or cDNA fragment. In those cases, it's always preferable that you go for low temperature for a longer amount of time. But for your regular cloning that you're doing in the lab, even 25-degree for 1 hour or 16-degree 4 hours, it's good enough. Anyway, the ligase, that the company from which you buy the ligase, they also prescribe that, okay, set up the ligation reaction at this particular temperature because they have standardized in their labs.
Okay, ma'am. And another question was, ma'am, can RNAs degrade the single stranded DNA?
No, it depends. No, RNAs will not degrade because they are specific for RNA nucleotides only.
But ma'am, for the exonucleases DNase one, that the reaction condition was stated there on the PPT is that it should be RNase free. So why so, ma'am?
Again, say I'm not remembering that slide. So you're saying that in exonuclease 1,
Yes, ma'am.
which acts on single stranded nucleic acid, you are saying that it should be RNase free.
Yes, ma'am. It's there on the PPT on the slide.
Yeah, because see, for each of these exonucleases, in detail, it is given in the NEB brochure. So you'll have to go
Yeah,
through
this
it.
ma'am.
But
This
let me
one.
just, this one, na, exonuclease, oops, just a minute.
And next to next.
Okay. Here.
Next, yeah, this DNase one, ma'am.
So no, DNA is one. Why you want RNase free? DNA is one is always used if you have to remove contaminating DNA from a RNA preparation. Let's say you are isolating RNA. It's not always that you will completely miss out on DNA in your RNA preparation. There will always be some contaminating DNA because finally you are lysing the cells and then isolating your nucleic acid. So generally what we see that there is some contaminating DNA. So how do you remove that contaminating DNA? By adding DNase. If this DNase preparation is not RNase free, then it will cut the RNase also. RNA also, sorry.
Achha, okay, ma'am.
है ना, तुम्हें RNA तो बचाना है ना, because you have to work on RNA only in that particular case.
Yes, ma'am.
Okay, guys. Then bye bye.
Bye.
Yeah. Take care.
Okay.